Dear students, let me talk about the generation of antibody diversity. Introduction. The effectiveness of the specific immune response is dependent on its ability to distinguish different antigenic determinants or epitopes. The immune system is exposed to an enormous variety of antigens in the form of infectious agents or chemicals and may be able to respond to such a challenge. However, the number of genes required to code for every possible antigenic determinants would be enormous. Over the years, many theories have been proposed to explain the range of epitopes recognized by antibodies. Two particular theories received the most attention and have been given greatest credence. The germline theory proposes that there must be a gene in the germline to code for every antibody variable's region and that these genes arose by duplication during evolution. By contrast, the somatic mutation theory proposes that only a small number of variable regions genes are present in the germline and that the product of these genes may vary greatly due to point mutation or recombination during lymphocyte differentiation, thus giving rise to a unique antibody repertoire in each person. Neither the germline nor the somatic variation proponents could offer a reasonable explanation for this central feature of immunoglobulin structure and diversity. Dreyer and Bennett proposed the two-gene model. Dreyer and J. Bennett suggested in their classic theoretical paper in 1965 that two separate genes encode a single immunoglobulin heavy or light chain, one gene for the variable region and the other for the constant region. These two genes must somehow come together at the DNA level to form a continuous message that can be transcribed and translated into a single immunoglobulin heavy or light chain. They propose that hundreds or thousands of variable region genes were carried in the germline, whereas only single copies of C region class and subclass genes need to exist. Tonegawa and Hozumi showed that immunoglobulin genes undergo rearrangement. In 1976, S. Tonegawa and N. Hozumi found the first direct evidence that separate genes encode the V and C region of immunoglobulins and that the genes are rearranged in the course of B cell differentiation. In 1987, Tonegawa was awarded the Nobel Prize for this work. Selecting DNA from embryonic cells and adult myeloma cells, cells at widely different stages of development, Tonegawa and Hozumi used different restriction endonucleases to generate DNA fragments. The fragments were then separated by size and analyzed for their ability to hybridize with radiolabel mRNA probes. Two separate restriction fragments from the embryonic DNA hybridized with the mRNA, whereas only a single restriction fragment of the adult myeloma DNA hybridized with the same probe. During differentiation of lymphocytes from the embryonic state to the fully differentiated plasma cell stage, the V and the C genes undergo rearrangement. In the embryo, the V and C genes are separated by a large DNA segment that contains a restriction endonuclease site. During differentiation, the V and C genes are brought closer together and the intervening DNA sequence is eliminated. Multigene organization of immunoglobulin genes. The kappa and lambda light chains and heavy chains of the immunoglobulin are encoded by separate multigenes family situated on different chromosomes. In germline DNA, Each of these multi-gene families contains several coding sequences called gene segments separated by non-coding regions. During B cell maturation, these gene segments are rearranged and brought together to form functional immunoglobulin genes. The kappa and lambda light chain families contain V, J, and C gene segments. The rearranged VJ segments encode the variable region of the light chain. The heavy chain family contains V, D, J, and C gene segments. The rearranged VDJ gene segments encodes the variable region of the heavy chain. C gene segments encode the constant region. Each B gene segment is preceded at its 5' end by a small exon that encodes a short signal or leader peptide that guides the heavy or light chain through the endoplasmic reticulum. The signal peptide is clipped from the nascent light and heavy chains before assembly of the finished immunoglobulin molecule. Allelic exclusion ensures a single antigenic specificity. B cells, like all somatic cells, are diploid and contain both maternal and paternal chromosomes. Even though a B cell is diploid, it expresses the rearranged heavy chain genes from only one chromosome and the rearranged light chain gene from only one chromosome. The process by which this is accomplished, called allelic exclusion, ensures the functional B cells never contains more than one BH, DH, JH and one BL, JL unit. This is essential for the antigenic specificity of the B cell because the expression of both alleles would render the B cell multispecific. The phenomenon of allelic exclusion suggests that once a productive BH, DH, JH rearrangement and a productive BL, JL rearrangement have occurred, the recombination machinery is turned off so that the heavy and the light chain genes on the homologous chromosomes are not expressed. Generation of antibody diversity. There are 7 means of antibody diversification identified in mice and humans. They are multiple germline gene segments, combinatorial B-D-J joining, junctional flexibility, P-region nucleotide addition or P-addition, N-region nucleotide addition or N-addition, somatic hypermutation, combinatorial association of light and heavy chain, multiple germline gene segments. There are numerous germline V, D, and J gene segments which contribute to the antibody diversity. The number of functional B, D, and J gene segments in the germline DNA of one human individual reveals to have 51 VH, 25 DH, 6 JH, 40 V kappa, 5 J kappa, 31 V lambda and 4 J lambda gene segments. In addition to these functional segments, there are many pseudogenes. The immunoglobulin loci of other individuals might contain slightly different numbers of particular type of gene segments. In the mouse, there appears to be about 85 VK gene segments and 134 VH gene segments, 4 functional JH, 4 functional JK, 3 functional J lambda, and an estimated 13 DH gene segments, but only 3 V lambda gene segments. Presence of such large number of gene segments aid in production of large number of antibodies with different specificities. Combinatorial B-D-J joining. The contribution of multiple germline gene segments to antibody diversity is magnified by the random rearrangement of the multiple gene segments of antibodies in somatic cells. In humans, the ability of any of the 51 VH gene segments to combine with any of the 27 DH segments and any of the 6 JH segments allows a considerable amount of heavy chain gene diversity to be generated, which is about 8,262 possible combinations. Similarly, 40 V-kappa gene segments randomly combining with 5 J-kappa segments has the potential of generating 200 possible combinations at the kappa locus, while 30 V-lambda and 4 J-lambda gene segments allow up to 120 possible combinations at the human lambda locus. These are minimal calculations of potential diversity. Junctional flexibility. The enormous diversity generated by means of V, D and J combination is further augmented by a phenomenon called junctional flexibility. Recombination involves both the joining of recombination signal sequences to form a signal join and the joining of coding sequences to form a coding join. Junctional flexibility leads to many non-productive rearrangements, but it also generates productive combination that encodes alternative amino acid at each coding join, thereby increasing antibody diversity. The amino acid sequence variations generated by junctional flexibility in the coding join has been shown to fall within the third hypervariable region or CDR3 in immunoglobulin heavy chain and light chain DNA. Since CDR3 often makes a major contribution to antigen binding by the antibody molecule, amino acid changes generated by junctional flexibility are important in the generation of antibody diversity. P-region nucleotide addition or p-addition. After the initial single-strand DNA cleavage at the junction of a variable region gene segment and attached signal sequence, the nucleotide at the end of the coding sequence turn back to form a hairpin structure. This hairpin is later cleaved by an endonuclease. This second cleavage sometimes occurs at a position that leaves a short single strand at the end of the coding sequence. The subsequent addition of complementary nucleotide to this strand, p-addition, by repair enzymes generates a palindromic sequence in the coding join, and so these nucleotides are called p-nucleotides. Variation in the position at which the hairpin is cut does lead to the variation in the sequence of the coding join. Therefore, it is also important in the generation of antibody diversity. N-region nucleotide addition or N-addition. Variable region coding joins in rearranged heavy chain gene have been shown to contain short amino acid sequences that are not encoded by the germline B, D or J gene segments. These amino acids are encoded by nucleotides added during the DJ and B to DJ joining process by a terminal deoxynucleotidal transferase or TDT catalyzed reaction. Up to 15 N nucleotides can be added to both the DHGH and VH-DHGH join. Thus, a complete heavy chain variable region is encoded by a VHN, DHN, JH unit. The additional heavy chain diversity generated by N-region nucleotide addition is quite large because N regions appear to consist of wholly random sequences. Since this diversity occurs at VDJ coding joints, it is localized in CDR3 of the heavy chain genes and thus helps in generation of antibody diversity. Somatic hypermutation. Additional antibody diversity is generated in rearranged variable region gene units by a process called somatic hypermutation. As a result of somatic hypermutation, individual nucleotide in VJ or VDJ units are replaced with alternative, thus potentially altering the specificity of the encoded immunoglobulins. Normally, somatic hypermutation occurs only within germinal centers, structures that form in secondary lymphoid organs within a week or so of immunization with an antigen that activates a T cell dependent B cell response. Somatic hypermutation is targeted to rearrange V regions located within a DNA sequence containing about 1500 nucleotides, which include the whole of the VJ or VDJ segment. Somatic hypermutation occurs at a frequency approaching 1 in 1000 per base pair per generation. This rate is at least 100,000-fold higher than the spontaneous mutation rate in other genes. Since the combined length of the heavy chain and light chain variable region gene is about 600 base pair, one expects that somatic hypermutation will introduce at least one mutation per every 2 cell division in the pair of VH and VL genes that encode an antibody. Most of the mutations are nucleotide substitutions rather than deletion or insertions. Somatic hypermutations occur throughout the VJ or VDJ segment, but in mature B cells, they are clustered within the CDRs of the VH and VL sequences, where they are most likely to influence the overall affinity for antigen. Combinatorial association of light and heavy chains. In humans, there is the potential to generate 8,262 heavy chain genes and 320 light chain genes as a result of variable region gene rearrangements. Assuming that any one of the possible heavy chain and light chain genes can occur randomly in the same cell, the potential number of heavy and light chain combination is about 2.6 millions. This number is probably higher than the amount of combinatorial diversity actually generated in an individual, because it is not likely that all VH and VL will pair with each other. Furthermore, the recombination process is not completely random. Not all VHD or VL gene segments are used at the same frequency. Some are often used, others only occasionally, and still others almost never. Conclusion. In conclusion, I would like to state that the enormous diversity in antibody molecule is generated by various factors, out of which the multiple germline gene segment and combinatorial VJ and VDJ segment joining could contribute to only few million antibody diversities when we are producing billions of antibodies every day. As the very large number of new sequences created by junctional flexibility, P nucleotide addition and N nucleotide additions are within the third CDR, they are positioned to influence the structure of the antibody binding sites. In addition to this source of antibody diversity, the phenomenon of somatic hypermutation contributes enormously to the repertoire after antigen stimulation. Therefore, junctional flexibility, P and N nucleotide addition, and especially somatic hypermutation together make an enormous contribution to antibody diversity. All of this accounts for up to about 10 billion different antibody combinations.